Today we're going to talk about biological macromolecules. And sometimes you might have heard this, like if you're really into nutrition or exercise, you may have heard the term macros. They're typically referring to these biological macromolecules. Now, what are the biological macromolecules? Well, they can be grouped into four basic categories, and three of the categories you might be already familiar with. In fact, if you look on the back package of any sort of nutritional or food package, you can find three of the categories right there. So the categories of biological macromolecules you should know for this class are carbohydrates, so carbs for short, or sometimes called sugars, but again, there's more than just sugars for carbohydrates. Things like fiber are also carbohydrates as well. Then you have lipids and fat, or sometimes just simply called fats. But lipids is a general term for these group of chemicals that have a lot of carbon-carbon and hydrocarbon bonds. Don't worry, we'll cover that in a short while. And then there are proteins. F in the chat. This is why I prefaced it because it's been pretty dodgy lately. So this is why I wanted to see that. But hopefully we're back. So stream is back. All right, the last biological macromolecules we're going to talk about are nucleic acids. So nucleic acids, do you see these in the nutrition facts? Well, you don't see them in the nutrition facts because nucleic acids, even though through complex biochemistry they can become metabolized into energy, typically they're not metabolized into energy because you would pretty much have to use up your reserve of all of these three before you start burning the nucleic acids in your body. So nucleic acids, not a major energy source. They're still a biological macromolecule. They're just not on your nutrition label because they don't really contribute to your caloric intake and energy intake. Okay. So let's first talk about carbohydrates. Let's talk about that first category. So carbohydrates, well, there are many types of carbohydrates, but what are the ones I want you to know? So the carbohydrates I want you to know are monosaccharides. So monosaccharides refers to one. And then there are many types of monosaccharides, but these are ones you should definitely know. So things like glucose, fructose, and galactose. And then you also have disaccharides. So disaccharides means two. So disaccharides referring to two simple sugars in monosaccharides joined together. So examples of that we have here. Now, there are many types of disaccharides, but the lists I'm keeping here or showing on this screen here, these are the ones you should have. And polysaccharides, poly mean many, and then so you have things like glycogen, starch, and cellulose. Those are examples of polysaccharides. So what about the structures and properties of glucose? So with glucose, what we have are, so here we have glucose, the structure of glucose, and actually, so this isn't an organic chemistry class, so I'm not going to, but often you might see with chemical structures, they just have like sticks and lines, but they don't say exactly what atoms are at these angles. So whenever you see this kind of like stick and line, and you see an angle like this, it's typically, especially when you're talking about biochemistry and cell biology, they're implying that carbon is here. So actually each of these angles here has a carbon atom over there. And also hydrogen atoms as well, but that's a little too organic chemistry. But what am I getting at here? So remember those polar bonds, those polar covalent bonds, and how oxygen bound to hydrogen makes oxygen a little negative and hydrogen a little positive? Well, this is where we have, so think about this, you have all these polar covalent bonds. So with all these partial charges on different atoms of glucose, the water is very attracted to that. Now, glucose overall has no charge. It's not an ion. But it's possible to have an overall molecule that has no charge, but different atoms on it have slight charges due to those polar covalent bonds. Okay, so then we have two monosaccharides, we have two carbohydrates here. So the cool thing about carbohydrates is that you can take simple carbohydrates and join them together and break them apart. Here we have two simple carbohydrates, glucose and fructose, and what happens is that you can do a reaction that joins them together. Now the cool thing is that you notice that we have this oxygen and hydrogen on both of these molecules coming together. but the end product only has one oxygen. So what happens to the rest of the atoms? Well, actually, those two hydrogens and one oxygen comes off of these structures. And hey, what has the chemical structure H2O? Hey, water does, right? So this is what we call a synthesis reaction, dehydration synthesis reaction, because you have water being produced as a result of this reaction coming together. Yep, so this is sucrose, so table sugar, or common sweetener. So table sugar is actually made from two smaller carbohydrates being linked together to form what we call a disaccharide. So sucrose is bigger than glucose, but it's made out of glucose and fructose. Now, there are many types of disaccharides, but these are the ones you definitely should know, especially if you're continuing on to PHYL 142, because there are special enzymes that help to catalyze these reactions. So glucose plus glucose makes a disaccharide we call maltose. So then, over here, glucose plus fructose, as we mentioned in the previous slide, that makes sucrose. And then glucose plus galactose makes a probably familiar sounding carbohydrate called lactose. So a common thing that trips up people think that, oh, galactose, because it has a longer name, is the disaccharide, and lactose is this monosaccharide. Lactose is actually bigger than galactose. So lactose is the disaccharide, galactose is the monosaccharide. And yes, thank you to people in the chat who are helping things with tech support. Again, this is why I go into work, because UH Wi-Fi connections are and Ethernet connections are way better than home connections and Spectrum. Okay, so back to carbohydrates. So we have one single carbohydrate, a monosaccharide here. So that's, again, one is a prefix, that prefix mono means one. And then when you have two monosaccharides joined together, that di prefix, di, refers to two carbohydrates. Now, when you have more monosaccharides linked together to this larger structure, then you have many saccharides. So there's another useful prefix that you see all the time in medical and scientific terminology called poly. So whenever you see something saying poly, that means many. So if you have many carbohydrates, more than two, let's just define it as that, more than two carbohydrates linked together, you have a polysaccharide. There are trisaccharides, but let's just say three or more. Yeah, two or three or more, you have a polysaccharide. So these are polymers. So the cool thing is, like, this term polymer, you may have heard this if you've taken a chemistry class before. But polymers, what you notice is that you have one monosaccharide and you keep on daisy chaining and having this repeating structure. So that's what we have there. So a polymer is a chain of any type of molecule, but you keep on repeating this link and joining them together through chemical bonds, so they form a larger and larger structure. So what we have here is a single person, and this is in the before times. So in the before times, you can hang out together, but what if you had a polymer of people? Well, you would have a repeating structure that has multiple single units in a repeating chain over here. So what we have here with all these people gathering together in the before times is a polymer of people. Okay, so then what we have here is that carbs, so carbohydrates, proteins, and nucleotides, they can form polymers. So many of these biological macromolecules, they can form these repeating structures theoretically until infinite, but again, your cells are finite, so they're not infinite. But they can repeat these structures over and over to go from a simple molecule and then build multiple bonds until you have bigger and bigger molecules. So fatty acids and lipids, these aren't exceptions. They can form complex molecules, but they don't typically form super long, potentially infinite chains of molecules and fatty acids or lipids. Now, what I'm getting at is that these biological macromolecules, they can join together and form bigger, more complex molecules. So an example of this is starch. So starch, here we have a single chain of monosaccharides together. So what you have here is that you have this monosaccharides forming a long chain. So this linear chain of monosaccharides In this case, we're talking about the long chain or single chain of glucose. It's called amylose. And then there's also a branched version of this. So polymers aren't necessarily just linear in a single chain that doesn't branch. Polymers can sometimes also branch. And in the case of amylopectin, is that you can have multiple chains of these carbohydrates branching and extending off into form complex structures. Now, glycogen, so you don't produce starch. Plants produce starch, but glycogen is like the starch of our own bodies. So your body is actually able to take, if say your body has too much glucose flowing around your bloodstream, does it have to just burn up that glucose immediately, or can it store it for later? The great thing about glycogen is that this is a one way your body can store the energy, or actually can store glucose molecules for later on. So things like your liver and your skeletal muscle, they're actually able to hold on to a limited amount of carbohydrates by making glycogen. Now, the glycogen, it branches even more than starch, and I know it's not very exact, but that's like a general difference between glycogen and starch. What you have are all of these carbohydrates, all this glucose linked together to form a branch structure. And the cool thing is, like, say you're burning through your energy and you actually want to release more glucose into your bloodstream that you've previously stored up. So the cool thing is that you're actually able to break apart glycogen and release glucose, so you're able to use that energy for later. Okay, so let's do our first top hat question. And yes, so our join code is here, and this is also the same code for our classroom as well. So that's what we have over. So I'm going to jog your memory. And I'm going to add a little time because I know I'm on my slower home network, so I'm going to give more time on this. And again, if you're joining the broadcast for the first time, questions asked during the live chat do not count for or against you. They're just for you to kind of test your knowledge. So here we have a reaction. Glucose plus glucose are the reactants, and maltose is the product. What terms best describe this reaction? And you should be able to select multiple answers. Yes, and that's a birb. That is my cockatiel, Mr. Peanut, down there, and he is whistling up a storm. And I'll get him later because right now he's like hiding under my desk. But all right. Okay, so what the responses are. So let's see what people said and what's the correct answers. Well, these are the correct answers. So there's more than one possible correct answer, but it is an anabolic reaction. Why? Because you're building a larger structure, and it's also a synthesis reaction because you're synthesizing a larger molecule and bringing molecules together versus breaking them apart. And let me see if we can get here. Say hi to the class. And actually, it's really funny. He hatched in 2004, so he's almost as old as some of you, or possibly older than some of you. So yep, say hi. Yeah, he's like, nah, I don't like the ring light. Okay, so if you hear weird noises in the background, that's due to the birb. Okay. All right. Okay. Okay, let's go on to our next molecule. So then we have fats. So then lipids, also known as fats. So these include fats, oils, and waxes, and things like this. So this is an example of fats, right? You have protein here, but all of this white stuff is fat. So if you've seen those really, really expensive Japanese, or I think now other people have gotten into the Wagyu business. But yeah, you have all this marbling, all that white stuff is fat, right? And then if you say you're a vegan or vegetarian and you don't eat meat, well, what's another good source of healthy fats are avocados. And yes, avocados are fruits as well. And then you also have things like oils, like I think this is olive oil because you have olives and all that. You know that delicious stuff they give you before, like California Pizza Kitchen, and then you can't resist all that, like the bread and oil. So those are also fats as well. And butter, like A lot of people love butter as well. So these are all examples of fats, oils, and waxes. Now, do these dissolve easily in water? Like, can you just throw these in water and mix them up and have an even suspension? Probably not. So that's another property, general property of lipids, is that they tend to not be water soluble. They tend to be hydrophobic. So this is another general property of lipids, and they tend to have something called long hydrocarbon backbones. And you might be like, whoa, what's a hydrocarbon? Well, just break that down. So you have hydrogen plus carbon. So these molecules that are lipids, they tend to have a lot of carbon-carbon and hydrocarbon non-polar covalent bonds, meaning that they're covalently linked, but they don't have those partial positive and negative charges you see with a polar covalent bond. So here's an example of one of the lipids, and this is a fatty acid. So this fatty acid, you might be like, what am I looking at this here if you've never had a chemistry class? Well, what we have here is like a space-filling model of a molecule. So the thing is that these fatty acids, they tend to have a typical fatty acids. There are many types of fatty acids, but in general, they have two main parts. They have a long carbon and hydrogen chain and a carboxyl group. And you might have noticed I tend to bold some words and I tend to italicize some words. So bold words are things I think you should definitely add to your vocabulary for this class. Things that are italicized, I tend to italicize them. Like, I probably won't go into detail in this class, but if you're taking further classes like organic chemistry or biochemistry, these are important topics to know later on in your academic career. So back to this little molecule we're looking at here. So we have a blue, so blue represents hydrogen, and then black is like pretty consistent among depicting molecules this way, and red is oxygen. So what we have here is actually a lot of carbon-carbon and hydrocarbon bonds. But here we have oxygen, and hey, we have oxygen, again in red, linked to a blue hydrogen. So this is actually a polar covalent bond, but all these bonds here are non-polar covalent bonds. And so this general part right here, so this has carbon, oxygen, oxygen, hydrogen. So this part right here is what we call a carboxyl group. So it's mostly nonpolar, except for that carboxyl group, because again, it has that polar covalent bond. Now, how does that relate to, why do we care? Well, it relates to hydrophobic and hydrophilic properties. So with this part right here, this carboxyl group is actually very attractive to water. So again, if we have this slightly negative oxygen atom linked to a slightly positive hydrogen atom, it's going to attract water molecules to this part. But what happens if you have a water molecule trying to associate with this part over here? Well, the thing is that none of these have a real dipole or partial electric charge. So this water molecule is not going to be attracted to it at all. So this part actually repels, or actually water, I should say that there is no real attraction between water and this part of a fatty acid. So overall, a fatty acid is mostly hydrophobic, meaning that water isn't really attracted to it, and vice versa, where only a small part of it is actually attracted to water. So fatty acids may be saturated, and this is again, if it's not organic chemistry class, so this is what I expect you to know at this point. If you're going to nutrition, you might want to know this, but saturated with single bonds or unsaturated with one or more double bonds. So there's also further things like monounsaturated fat, polyunsaturated fat. What does that refer to? Well, that refers to different arrangement of bonds. So sometimes you might have a double bond between carbon atoms, sometimes you might have a single bond. Again, I think that's a little beyond the scope of this class. Maybe we get into that when we talk about nutrition. But in terms of chemical structures, that's more for like your more advanced chemistry classes. Okay. All right, so then let's talk about glycerides. So think about, why do we have to talk about glycerides? Well, glycerides and glycerol itself is not a lipid. But the interesting thing is that it's very important for how you're able to store and transport, and also store and transport lipids in your body. So glycerol is actually hydrophilic, but this is why we have to talk about glycerol. There are many types of, there's a type of lipid in your body called triglycerides, And what they are 3 fatty acids plus a glycerol. Common thing that trips people up is that they think the triglyceride has 3 glycerols and 1 fatty acid. The tri part refers to fatty acids, the glyceride refers to glycerol. So they're also sometimes called triacylglycerols, but in this class I tend to just say triglycerides. So say your parents or an elderly relative says like, oh, my triglycerides are up. This is what they're referring to. They're referring to this type of lipid in their blood. So here's glycerol, and again, glycerol itself is not a lipid, but when you have a triglyceride, you actually take 3 fatty acids, and then you synthesize them together with the glycerol, and now you have a larger molecule called a triglyceride. So again, this is why it's important to know what glycerol is, because it kind of folds, it's kind of like a scaffold that holds up to 3 fatty acids together. So this entire structure here is actually a triglyceride. But again, the tri refers to the 3 fatty acids that are linked to 1 glycerol molecule. And there's another major category of lipids called steroids. So steroids, whenever people think of steroids, they think of something like this. But they're probably thinking of anabolic steroids. And anabolic steroids are a type of chemical we call steroids. So most steroids have this classic structure. So what they have is all these numbers, red here, numbers here. Now, at this level, I don't expect you to know all these numbers right there and the exact chemical structure of a steroid. Actually, all those red numbers refer to a carbon. So you have all these carbons that form the structure of a steroid. So they are lipids, and they have a bunch of carbon-carbon and hydrocarbon bonds, and they tend to be nonpolar and hydrophobic. So why are they important? Well, they're not only important, if you're into bodybuilding, they might be important, but they're also important for lipid metabolism as well, and also very important in hormones. So there are a category of hormones we call steroid hormones. It's not just testosterone, but you also have things like estradiol, which is a type of estrogen, progesterone, cortisol. Those are all types of steroids in your body. So steroids, they all kind of look like this. So here we have cholesterol and estrogen and testosterone. So notice that estrogen and testosterone, actually I clarified here, estradiol, looks like a cholesterol, right? And what do they all kind of have in common? They have this 4-ring structure. So this is why they're all so labeled A, B, C, D. So it's funny, like I was able actually to teach one of my little cousins how to do this, like how to recognize a steroid. I just say it's 3 hexagons linked to a pentagon. I mean, this is like this ABC, this 4-ring structure. If you know geometry and your shapes, you're able to kind of recognize the general layout of a steroid. So again, these 3 hexagons linked to a pentagon, that is a classic steroid structure. And all steroids, at least in your body, they're derived from cholesterol. So again, they all have that common 4-ring structure. Okay. So there's a special type of lipid called phospholipids. Now, this is a phosphate chemical group, and it's kind of hard to see here, but in this phosphate ion, it actually has a -3 charge. So one oxygen is neutral, but these actually have a charge of -1 each. Now, with this negative charge, it's very, very, very attractive to water because, again, water has those partial positive and negative charge. Since there's a negative charge on a phosphate group, the hydrogen of these water molecules are going to be very attracted to this phosphate group. Now here we have a phosphate linked to a nitrogen, or like a partial hydrocarbon, but another polar group. So this part right here, this overall chemical structure, when you have a lot of ionic charges or polar covalent bonds, that's very attractive to water. Now, this part is going to be very hydrophilic because, again, you have either full or partial charges on these atoms. But when you have a phospholipid, you're also linked to, very similar to those fatty acids. Actually, fatty acids help to form this part of the phospholipid. So this is actually hydrophobic, and why is that? Well, this part right here is showing actually a lot of carbon-carbon. These are hydrocarbon tails. So they have a lot of those non-polar carbon-carbon and hydrocarbon bonds. Because they're non-polar, they don't really have an electric charge, therefore there's nothing for water to really grab onto. And so this is one way of showing, a more detailed way of showing a phospholipid, but you might see a more simplified version in your book. And this is kind of representing it. So they like to say that the part with the phosphate group and the glycerol is the hydrophilic head. And those parts that dangle off right here, those fatty acids, those are what they call the hydrophobic tail. So it's basically, in a single phospholipid, you can have a part that's more attracted to water and a part that's less attracted to water. So this is why we had to cover hydrophilicity and hydrophobicity. So again, hydrophilic, meaning that you have a part that likes to associate and bind with water. And then you have this part that's not really attracted to water over here. Now, the cool thing about phospholipids is that they're a special category of chemical, or actually, they get a special designation called amphipathic. So when you have like an amphitheater, it's like half, right? Half of a theater, half circle. So amphipathic is kind of saying that it's kind of falls between both worlds. It's not quite fully hydrophobic. It's not quite fully hydrophilic. So this phospholipid right here is actually both hydrophobic and hydrophilic. It just depends on the part of the phospholipid you're talking about. Identify this type of molecule. Or in other words, what descriptor best describe general descriptor, or can you summarize what these molecules are in one word besides complex? So the response is, seems like most of you said steroid or steroids, and yes, these are definitely steroids, and they're not carbohydrates. So carbohydrates are a totally different class. Steroids are a type of lipids, and triglyceride, no, it's not a glycerol attached to fatty acids. But yeah, some of you got really specific over here. And interesting question. Should we study the book more than the lecture? The book went really in depth. My advice is lecture first, and then the book to supplement your understanding of the lecture. Some people like a lot of detail, but I work from what I cover in lectures as a base. And don't forget the homework that comes out as well, because sometimes there's material that I think is better served studying on your own time, especially stuff that is like you have to practice over and it's more rote memorization. Me repeating the same thing over and over in a lecture won't really help you that way. So like memorizing or like recognizing the muscles, those are like, so lecture, I would say lecture, homework, and then the book to fill in the gaps. Back to our presentation. Yeah, because I know like the book, especially with the chemistry, they're going to go super duper deep in depth in some of the chemical parts, but pretty much what I cover with chemistry is what I think is most relevant and goes furthest in terms of your knowledge in this class. Again, I'll leave the details like valence shells and orbitals to your Chem 161 professor. Okay, last but not least, or actually not last but not least, but the last of the nutrition, the ones that add to your calories. So we're going to talk about proteins. So proteins, there are so many different proteins. They are the workhorses of your cells. They're forming a lot of the structures of your cells. They form the moving parts of your cells. They're most of their enzymes in your body. They're proteins. They do so much in your, so they're not just anatomy and structures, but they're also a lot about the chemistry and physiology as well. So proteins in general, they contain carbon, well, the organic molecules that contain hydrogen and carbon, but proteins contain carbon, hydrogen, nitrogen, and oxygen. So the basic parts, so remember how like monosaccharides, you can make a polysaccharide, like, or polysaccharides like starch or glycogen from monosaccharides? Well, proteins are made out of smaller units called amino acids. So there are many types of amino acids, but when you build along amino acids into a long chain, this is when you get a protein. So there are 20 standard amino acids, and that's all I expect you to know at this level. If you go on to maybe cell biology, I'm not sure if they teach it in bio, was it 265, 275? I forgot. But there is a special one called selenocysteine, don't worry about for this class. And I believe there's another one like in archaebacteria called like pyrrolysine. Don't worry about that one either for this class. But the ones that build proteins, the basic standard ones, there are 20 standard amino acids. So the protein structure, so what is amino acid? So we see a simple amino acid structure over here. And then what happens with the protein or actually polypeptides is that there are long chains of amino acids. So this is the same chemical structure that carbon, oxygen, oxygen, hydrogen you see with a carboxyl group in the fatty acid. But now it has this amine group, so it refers to this part right here. So this nitrogen atom, you don't find that in a fatty acid. You don't find that in like carbohydrates like glucose. But, or actually, let's rewind that part. So this is like, amino acids are different from fatty acids for sure. Now, amino acids, they all have this basic structure, but between different types of amino acids, they have something called a variable group or side chain, and it's represented as R. Now, it's not referring to an atomic element R, it's just saying that between different types of amino acids, you can have multiple atoms or sometimes just a single atom linked to this basic common groups you see in all amino acids. So this part, it varies. And here's a list of all the, oh, this one is comprehensive. It has 21 amino acids. Again, for this class, don't worry about selenocysteine. Actually, it has selenocysteine over here. Cool. But notice that this part over here on the top in these diagrams, it's all consistent. It's just that part that dangles down that makes the differences between all of these amino acids. Now we have to talk about peptide bonds. So here we have two amino acids, and notice that they're mostly the same. It's just that the side chain is different. Here we have a single hydrogen atom here, and here we have a methyl group. Now peptide bonds, these are what linked amino acids. So what we have here is that we have amine group over here, and it's going to have a synthesis reaction between amine and this amine group and this carboxyl group. So this part right here is going to actually pop off a water molecule. So remember, it's that dehydration synthesis reaction, like the one we saw between glucose and fructose to make sucrose. So it's very similar that you have the synthesis of a larger molecule from two smaller molecules. So this bond right here, so there are many bonds in this structure, but this specific bond that happened between these groups, that's what we call a peptide bond. So peptide bonds, these are new bonds between these amino and carboxyl groups, between this carbon and nitrogen. And so this is my definition. Like, you can talk about the biochemical definition, but this is all what I want you to know for this class. So this is just like, basically, peptide bonds are what join amino acids. That's pretty simple, right? So if you have many, so remember that poly prefix, that root word? If you have many peptide bonds together, this is what you have with the long chain of amino acids. So if you have a long chain of amino acids linked with many peptide bonds, you have a polypeptide. So again, 20, and not including selenocysteine, you have 20 different amino acids. So if you have all these amino acids with different side chains, but the thing is that between each amino acids, you have a peptide bond. So if you have many of these peptide bonds linking a long daisy chain of amino acids, you have a polypeptide. So again, this refers to some, and there's some other small terms regarding peptides, but don't worry too much at this point. So again, what I'm getting at here, so if you have a polymer of amino acids, so you take these basic building blocks of amino acids, link a lot of them with peptide bonds, you have a polypeptide. So a polypeptide is a polymer of amino acids. Now, the thing is that with the proteins, like this is, again, very complex chemical structure, showing that all these angles here, they would be carbon, but you can see that you have all these atoms linked together. So this is actually the structure of what a probably familiar hormone and maybe medicine if you have a diabetic, if you're diabetic or know someone who has diabetes. Well, this is insulin, and it has a very complex chemical structure if you look at the individual atoms. So when you talk about the protein primary structure, you're talking about how it looks like a sequence of amino acids if you stretched it out into a long chain. So that's what we have here. I like to use an analogy. This is a molecule of hair, But when you're talking about, ask someone to show you a picture of hair, are they going to show you a super zoomed in version of that? So think of it this way: a protein's primary structure is like that sequence of amino acid, but it's like the highest resolution, most detailed, probably more information than you need when you're talking about the protein. So here's a secondary structure of a protein. So remember, proteins are made out of polypeptides, and polypeptides have these long lengthy structures. There are all these amino acids linked together. So here we have an alpha helix, and here's a bunch of, so this ribbon is representing a chain of amino acids, but you can also twist these amino acid chains into shapes. So what we have here is kind of like this alpha helix is kind of like rolling, like if you have seen someone with dreadlocks, they kind of roll their hair together to form a bigger structure. So that's what you're kind of doing with the secondary structure of a polypeptide or a protein, is that you're kind of like folding it and making more complex structures over from a single strand. And here's the beta sheet. That's another example of that. Now, tertiary structure, well, this is when you're taking those basic structures and making even more complex structures. So you're folding and making a 3D arrangement. So here we have an alpha helix, like in that secondary structure. So I like to think of it this way. Like in those previous examples, I showed you like that strand of hair. I also showed you that like different ways you can make style strands of hair. Well, if you have multiple of these structures linked together, you can have fancier and fancier structures. So as you go from primary to secondary to tertiary, you're able to build more and more complex structures. So again, and then with quaternary structures. So here's an example of a protein that has a quaternary structure. So common pitfall when you're first learning about proteins is you think all proteins are just one polypeptide. So some proteins actually have multiple polypeptides linked together to form one functional protein. So in a single molecule of hemoglobin, you actually have 4 protein chains. So there are actually 4 separate polypeptide chains that are folded together, and they form a larger structure called hemoglobin in this case. So it's not just one protein. All proteins have just one polypeptide. Sometimes proteins, you actually need to bring multiple polypeptides together to form a full protein molecule. So to extend my hair analogy, what would be a hair analogy for quaternary structure? Well, in the tertiary, we saw those complex hairstyles. Quaternary structure is like if you took 2 tertiary structures and put them together. So like this. So it's not just hemoglobin, but like collagen as well. Collagen is actually all of these molecules over here. These are 3 polypeptides linked together and kind of twisted together and braided together. So that's why I'm kind of drawing that analogy and the difference between tertiary and quaternary. If there's more than one polypeptide involved, it's a quaternary structure. All right, and then last but not least, so we already covered the carbs, lipids, and proteins, the ones that you can find on your nutrition label. Now let's talk about nucleic acids. So nucleic acids, they're the ones that aren't on your label because they don't really contribute to your caloric intake and energy. Now, nucleic acids, here's one that might seem very familiar to you. So this is DNA, and we notice that notice that it kind of repeats over and over and has this kind of polymer-like structure. So these are long chains of nucleotides. So nucleic acids are polymers of nucleotides. This is why they have that kind of repeating structure you see with this double helix in DNA. So DNA stands for deoxyribonucleic acid. So that D is very important, and then nucleic acid is the N and A. Then this is the very, very famous RNA. So that mRNA you've been hearing about with those vaccines, they're a type of ribonucleic acid. So they're related, but not the same. They're very similar, but DNA and RNA aren't exactly the same. So with RNA, typically when you find RNA in your cell, unless it's like tRNA or rRNA, but when we talk about the mRNA, it tends to be a single strand. So here we have RNA, a single strand of that, and whereas DNA, notice that you actually have 2 of these, what we call a sugar phosphate backbone, and it's represented by these gray ribbons over here. So this is why you might have heard the term double helix, because compared to RNA, which only has 1 backbone, Notice that DNA has this two backbones twisted together. So that spiral shape is what you call a helix. So that's why they say DNA has a helix, double helix shape. And one other difference between DNA and RNA is that notice that DNA and RNA, they can include these three nucleobases here, cytosine, guanine, and adenine. Same in both. But DNA has thymine and RNA has uracil. So these are the basic nucleobases. So these are the parts that can change between the different steps of or along a chain of nucleic acids. So DNA has this set of 4, and there is overlap. RNA does have adenine, cytosine, guanine, but thymine is with DNA and uracil is with RNA. Now, what happens when you take a bunch of these nucleotides and link them together? Well, this is when you get a nucleic acid. So I like to think of it this way. Why do those nucleobases and those 4 different types, depending if it's DNA or RNA, why does it matter? I like to draw this little analogy between a nucleic acid and like a charm bracelet. With the charm bracelet, you have a basic structure that holds it all together. So this sugar phosphate backbone of a nucleic acid is going to be the same regardless of what's attached to it. This backbone repeats over and over and over again, just like this chain on this little friendship or charm bracelet over here. But the parts that dangle off, those parts are different. They have different letters, and these letters encode information. So just like this little friendship bracelet has information, has a message, same with the nucleobases that are off a sugar phosphate backbone. They have a message. And yes, those are birb noises as well. Okay. And he's like, I can't stand you having all the attention. I want some attention too. Okay, so then macromolecules and polymers. So you have different types of polymers. You have polysaccharides. These are polymers of monosaccharides. Polypeptides, they are polymers of amino acids. And nucleic acids, these are polymers of nucleotides. Now, do fats and lipids form polymers? Well, are there lipid polymers? Well, triglycerides, even though you do have a synthesis reaction when you take 3 fatty acids and link them to glycerol, this is a synthesis reaction. It is an anabolic reaction, but since there are only 3 oxygen molecules on a glycerol molecule, then you're only limited to 3 fatty acids. So triglycerides are not polymers. If you could have a polymer, then it would look like this. Yeah, he wants to teach everyone how to whistle. Okay, so then this doesn't happen in your body. So with the glycerol molecule, you can have up to 3 fatty acids, but you can't have fatty acids binding over and over and over and over again. So fatty acids do not form polymers in physiological conditions. Now steroids, if steroids could form polymers, they would form polymers like this, but they don't do that. So steroids, another type of lipids, these do not form polymers. So pretty much 3 out of the 4 biological macromolecules can form polymers, but lipids cannot form polymers in physiological conditions. So I have this little chart that kind of summarizes all of this in your slides. So this is kind of like what I want you to know at this point. You can read the textbook if you want more details about this. And there are other types of macromolecules as well. So you have glycoproteins. So glycoproteins, well, this is actually a very useful prefix over here, glyco. So remember how they have that carbohydrate called glycogen? Well, glyco typically refers to some sort of sugar. So when you have a glycoprotein, you have a carbohydrate linked to a large protein. So these include there are many types of glycoproteins. Don't worry about memorizing a list of them right now. Just know that sometimes you can actually link different types of biological macromolecules to other categories as well. And then you have something called proteoglycans. And these are different arrangements, but they tend to have a polypeptide core, and then they have a large polysaccharides branching off from them. So these are very important in making like fluids in your body thick or holding on to water in parts of your body. And you also can have like glycolipids. So glyco, again, useful root word meaning referring to sugars or carbohydrates. So this is going to be a sugar molecule linked to a lipid. So glycolipids, these are complexes forming from both a hydrophilic carbohydrate and a hydrophobic lipid. So you actually find these not, so your cell membranes, they have phospholipids, but they also have these glycolipids as well. Okay, so let's end the lecture with some top hat questions. Yeah, that's the thing. He just does his own things these days. I don't know how you convert between cockatiel years and human years, but okay, let's see what people said. So which of these tend to be hydrophilic? So these tend to be hydrophilic. So pretty much the 3 of the 4 main categories, like carbohydrates, they definitely tend to be hydrophilic. Otherwise, if you just threw sugar into water, it wouldn't dissolve, right? Proteins, there are some hydrophobic proteins, but in general, proteins tend to be hydrophilic because, again, you have proteins all over in your body, so you need proteins to associate with water. Nucleic acids, so the inside of your cells, a little spoiler for the next few lectures, are mostly water-based. So nucleic acids like DNA and RNA, they definitely need to dissolve in water. And the fatty acids, remember fatty acids and the hydrocarbon tail of a phospholipid, they have those carbon-carbon and not hydrocarbon bonds, which are nonpolar, so they don't really have a charge to attract water. So these tend to be hydrophobic, and the rest tend to be hydrophilic. He is a little nut. Yeah, but yeah, I love. If you've ever had cockatiels, they're hilarious, and they have such personalities, as you can see. So I try to keep like multiple correct answers to a limit, but most of them tend to have one correct answer. All right, so to a little review. But if it does have multiple correct or select all that apply, it will tell you that select all that apply or multiple answers are allowed. But if it doesn't, then and you're only able to select one, select the best answer. And actually, it looks like I didn't select the best answer for this, but there is a correct answer to this one. All right, so let's see what people said. And yes, you are correct. It should say nucleotides, but yeah. Yep, so that's all the questions I have for today. And all right, say bye for now, Mr. Peanut. Okay, he's not happy having it. But yeah, thank you for putting up with like the connection issues earlier and the impromptu concert. Yeah, I'm not criticizing you. Okay. You'll get scritches. All right, see you everyone.